actually, thank you, Bar. So this is, we're now starting the recorded section of this presentation. Bar, if you could help me stay on track, I'm going to set go, but if I go over, by all means, unmute and pull me off screen. So with that, I'm gonna hit go. We'll go into the next slide. Sorry. Okay, ISCN. So the ISCN is an International Soil Carbon Network, and we created a data template in 2016 that was intended to cover all layer level information pertaining to soil carbon dynamics. So it was a very large data template. And as part of that effort, we had to think about how would we identify those layers. So they came up with this following sort of nested structure for it. So there was a site that is a geolocation containing a cluster of profiles that is constructed of depth interval layers. This was effectively dropped to be essentially a site which had a temporal and a geolocation, latitude, longitude in this case, that was associated with a particular layer. And the layer had an upper and lower bound. So this nested distinction between a site, a cluster, and a profile was effectively dropped when the data actually started being aggregated together. So that was something that was quite interesting. So things that the International Soil Carbon Network learned was that the geolocation, which is currently represented as a point-based latitude longitude with a datum. So what latitude longitude system are you using? Didn't really do a great job of incorporating uncertainty into it. People tried to do it with kind of a precision for the truncation for the latitude longitudes, but that was really easy to get lost when you started processing this through Excel or other data processors where you had that numerical precision just wasn't maintained. All right, I'm at 2 minutes. Sampling design and structure is not really well described in the data model. We need to improve that. Depth intervals needed 0 reference. That was not always recorded, should be. And time matters and can be different for the site and the layer level observation. So you need a time stamp on both of those locations. So on that note, moving on to Arden.
Hi, so aridec is a plant litter decomposition database focused on experiments carried out in arid lands specifically. For those, we included publications, and from those publications, from each publication, we created one entry. Each entry has a table with mass loss data as a time series, and then you can see we have another file, which is the metadata file, which is like a list where we included all study design and study site information that is relevant. And we have another table, which is the initial conditions table, in which we included all the initial traits of the plant litter used in the experiment. If you can go to the next. So lessons learned from aridec. We successfully stored data with different formats in our database, mainly text and tables. And these files can be used with most software, so it's pretty accessible. We also created an R package to help us read all different formats, all these different formats in the same workspace, and it's pretty effective so far. But I recognize it can be limiting for those who do not use R, we still have one major limitation, which is linking. We can link one mass loss time series to data in other files, pretty straightforward. except for when the entry has more than one species or site level in the experiment. For those cases, the approach is still pretty manual. It can be done, but it's not that simple.
Perfectly on time. Thank you. All right. Let's move on to Downwood Material.
Hi, I'm Erin Berryman. I'm with USDA Forest Service, Forest Inventory and Analysis, and I am based out of the Rocky Mountain region. So a lot of the stuff that I'm going to talk about does relate just to Rocky Mountains. FIADB, the database, has downwoody material litter and duff data for anyone to access. It's important to know some details of the methodology though, because this explains what the data look like when you try to access them. It's a transect tally method. We are not counting every single piece of wood on the plot. That's important to know. You'll see on the right side, we have 4 24-foot radius subplots that make up one plot. This is the classic FIA design here that we also measure all the trees and other stuff. Now the coarse woody debris and the fine woody debris, which is what makes up the downwoody material, are only measured if they intersect with any of those 2 transects that are on each of those subplots. And we do measure the diameter at the point of intersection, but everything else is just classed by diameter class. Litter depth and duff depth are also measured at each subplot, and then a density factor is applied to come up with the biomass per acre values. So if you want to access the data in FIADB, we do have a table that has the biomass and carbon values already calculated, but it is of course important to understand where exactly these data are coming from if you want to aggregate it at different levels besides the plot and condition. And next slide. So we have so many lessons learned. I couldn't fit them all onto this slide. We are in a state of transition, and this is going to affect any updates and the documentation ecosystem that we have. So the new database that we're working towards, hopefully it'll come out in the next 1 or 2 years. We'll have integrated machine readable metadata among other improvements. Right now, a lot of this information, unfortunately, is a little bit buried from the methodology standpoint. The downwoody material data is unique in FIA, and so it's not very plug and play. It really does require some understanding of the FIA plot design. We got data gaps, changes in methods that really make changes over time tricky. And you always have to keep in mind, as with everything with FIADB, specific protocols can change from region to region.
All right. International Soil Radiocarbon Database.
Great. So I'm Shane Stoner. I'm with the Max Planck Institute for Biogeochemistry. And yeah, we're talking about soil radiocarbon, which is a really powerful and cool metric for integrating a lot of processes that go on in soils and creating a mean radiocarbon value that can also then be split into a lot of a spectrum of ages, which I think Kate will talk about next. We took a lot of lessons from ISCN, including like the fact that time and profile are individual and important metrics that vary, with time and with location. Since radiocarbon is a radioactive isotope, there's important information that's required for the date of collection because those numbers can change over time, and this data needs to be corrected. And in order to interpret radiocarbon on a shorter time scale, integration with models is really critical. So we can go to the next slide. The International Soil Radiocarbon Database, which a few people on this call have been involved with. We use Excel as our main template, and we built an R package to integrate and kind of unify all of these different data streams. So we designed the template to be flexible to include data from a lot of different types of experiments. It's all linked at the publication level, and we have this kind of cascading metadata structured so that everything can be linked and compared across studies. We have a published version on CRAN and a development version on GitHub. We use both computer and human QA/QC. One thing I will say about making a spreadsheet template like this is that making changes in the future can be very expensive. So just think hard about your template design. Thanks.
All right, thank you so much. Okay, more ISRaD. Talking about fractionation schemes now.
Yeah, so another essential part of ISRaD, International Soil Radiocarbon Database, is its ability to absorb and integrate data on soil fractions. And why is it so important to combine radiocarbon and fraction data? That's because, as Shane said, radiocarbon is this great integrator of all the processes happening in soil. It can give you some idea of how quote unquote stable or persistent that soil carbon is. However, there is a huge difference between like the cycling or stability or turnover time, whatever phrase you want to use, between different fractions. So a fraction is just a subcomponent of a bulk soil. And there are, as you can see, kind of at the second layer, fractions are embedded within the bulk soil. And there's a lot of different methods that people have used historically and currently to fractionate soils. And so using all sorts of different properties of the soil, you know, based on whether or not it's in an aggregate, whether or not it's attached to a mineral surface, the size of the aggregate, the size of the mineral particle, its physical characteristics, its thermal characteristics, so how its propensity towards combustion. So there's all these different kind of approaches that we use to try to fractionate soils, and it's important for ISRaD to be able to have some way of comparing and comparing among those approaches and being able to look at the information, different information that they give you. Okay, next slide. As Shane already said, think about your template before you do it, because fractionation is really complex. And recently, Shane and a lot of other ISRaD members spent a good chunk of their winter break last year basically redoing and reclassifying the structure of the fraction portion of the template, because it wasn't able to effectively give them what they give us the information that they needed. So that's it.
Awesome, thank you. And next is this soil respiration database.
Hello, everyone. It's glad to be here to talk about SRDB, which is the acronym for Global Soil Respiration Database. It was first released by Lombardi and Thompson in 2010. And after that, the development team are working on keep updating this database. So what we did is every year we use the keywords soil respiration, soil CO2 fluxes, or soil effluxes in the Web of Science, and search the literature related with the soil respiration. And usually that returns lots of papers. So what we did is we did the first round of data filtering by reading the abstract and the title to do first round of filtering. Only those paper we think will report soil respiration field measurements will be used in the next step. So what we did in the next step is download the paper and then reading through the paper, and the data will be included in the database. And this database make up of 3 CSV files and also related description file, which is named as a txt file. And the core part of the data is SRDB data. And here we have 4 types of data there. The first group is record metadata. And then the second group is the site and measurement data. And then it's annual and the seasonal soil respiration fluxes. And then the last one is the ancillary pools and the fluxes. And the next slide, please. So what we learned from this database, the first thing is it's important to update the database regularly and timely. And the second one is don't try to including everything from the paper. And the third one is don't let the perfect to be the enemy of good. And the last one is important to standardize the data collection, and so it's easy for the user to participate. Thank you.
Excellent. Thank you. Okay, up. Open Soil Spec Library.
Okay. Hi, everyone. I'm here to talk about the Open Soil Spectral Library. It's part of the soil spectroscopy for the Global Good project. And here we have a representation of the soil model, soil data format. We have soil site information, soil analytical data, VSNIR spectra, and also middle infrared spectra. All this data is linked with a common and shared identification column. And we have a representation of the MRR spectra in the right side. And with this project, we are gathering many soil spectral library public and licensed and distributed around the world. And we have, for example, for the MIR data set, some specific format like covering the 600 to 4,000 centimeters with a spectral resolution of 2 centimeters, and also formatting the spectra to log absorbance. That's what we are using. Please, next slide. So we have learned a lot of things, and there are different spectral acquisition protocols, different instruments, and we have to standardize, format this data for this open soil spectral library. And we are currently participating in a IEEE working grouping for defining and providing standards and protocols for soil spectroscopy. So from then we can try to calibrate and distribute like prediction engines. We can fit globally prediction models, and that's what I want to say. If you need more information, please visit soilspectroscopy.org.
Thank you. All right. You had 3 seconds left. Okay. Monet. Go to FTICR.
Yeah, I'm Maggie Bowman. I'm presenting today about MONET, which is the Molecular Observation Network. It's an effort being put together as part of EMSL and PNNL's 10-year vision for building a molecular observation network of soil properties, and that includes tomography for structure and FTICRMS for composition. One of the big things I wanted to highlight with talking about FTICRMS is that for each of the soils that we collect, we send out a standardized kit, and then we provide a standardized set of data collection and analyses, which allows it to be intercomparable. And we're working on building a comparable across time and instrument database with this. But as a result, we've had some challenges, and that's, standardizing how to measure organic matter in soils from a diverse set. So if you want to go to the next slide. One of the big challenges we've had is how much soil do we need? What's our solvent of choice and our solvent ratios? How are we going to clean up these standards? And we're working on having a database that will be available. I believe it's planning this fall or winter, that will have the standardized output molecular characterization, as well as some more simpler values like the means of these distributions with space. And so we're really trying to make a mass spectrometry incorporated into a database, which hasn't been done at this scale yet before. I think that's all I had. Sorry, I probably have time.
10 seconds. So
okay, perfect.
Good timing there. All right. Okay, last but not least, Ag Evidence.
Hi, everyone. I'm Leslie Atwood. I'm with the Nature Conservancy. And the Ag Evidence database and web visualization tool features what Kathe has informed me is rescued peer-reviewed data on the impact of agricultural practices on crops and the environment. So it's a giant database covering a lot of topics. We first developed our relational database around corn soybean management in the US Midwest, but we've recently expanded it into diversified cropping systems in Kenya, and we hope to add more geographies as we move forward. But for the US Midwest, we targeted 4 management practices: cover crops, nutrient management, pest management, and tillage. So for each of those practices, we created a hierarchical group to capture the gradient of disturbances, because each specific practice, of course, can affect our soil physical, chemical, and biological aspects to varying degrees. And we also wanted to pull together similar practices that aren't as widely known, such as like vertical tillage and stubble mulch, and to recognize groups like conservation tillage. In terms of the measured outcomes, we collect everything that is reported in each paper, which is a little crazy, yes, but we did it and we're doing it. So we have grouped those outcomes into outcome groups that are recognizable: climate mitigation, crop yields, economic, soil properties, pests, soil nutrients, and water quality. But then within each of those outcome groups, we break that again down into subgroups to get down to the nitty gritty. So you can go from other soil properties to soil structure to bulk density down to the actual data that we collect out of the paper. And that's all you can navigate that through our web visualization tool, but it's also embedded in our database. Okay, I think, oh yeah, and we were able to do that because we have a highly flexible database structure. And that really, I have to say, props to the National Center for Ecological Analysis and Synthesis. They really helped us set up our database so that it would work for us. We didn't really know we were going to do a web tool at the end, but it's worked out perfectly. Okay, that's what I've learned. And try Colander if you're doing a literature review. It's very helpful.
All right, thank you so much. Okay, that was fast. 2 slides is not enough for any of these individual projects. This represents, all of these individually represent a huge amount of work. So thank you all for coming and trying to cram everything into 2 slides for me. I dropped into the chat the 1-page document that we've also asked the presenters to provide if you want a little bit more details. But right now we've got 5 more minutes for clarifying questions before we go into breakout rooms to talk about what we just presented, that will then go around and have a deeper discussion on. Because we do have over 30 people in the breakout room, I'm asking people to either put their clarifying question in the chat or raise your hand if you have something you want to ask before you go into breakout room to talk about what we just heard. And if no one says anything for the next minute, we'll just roll into the breakout rooms, and then we'll come back and we'll have a discussion. So clarifying questions.
Question on the breakout rooms, are we going to different rooms for each speaker that we want to know more about, or is it just a general breakout room?
It's going to be a general breakout room. I'll put you 4 to 5 people in a breakout room. And then what your task is going to be is to come up with a recap. Actually, since before I've got another slide on this, but if you want to,
fine, that you can talk about it later. I do have a clarifying question for Leslie. Okay. Just wondering on the data, anything about microbial processes? I heard a lot of physical properties. Is that pretty much what you focus on?
We include anything that's reported. So microbial processes, soil fauna, everything. Yep, it's all there.
Thanks.
Yep, you're welcome.
I have another question for Leslie. Who's doing all this data entry?
Hello. It's me. Yes, it's quite the process.
any more questions? All right. In that case, okay, I'm going to, got 10 minutes into these breakout rooms, and you have 2 charges in your breakout room, and there will be a report out afterwards. So be sure you report, you appoint a recorder. And your 2 charges are, what are some of the common themes across these data collections? Are there anything that seems to be sort of true across that we can maybe tease apart as a potential start for a common resource. And then what questions did these sets of presentations raise for you? So what are you curious about? What do you want to know more about? When we come back, we'll have 15 minutes and we're going to do this report out in 2 rounds. The first round is to sort of gather new points. So what's something that was discussed in your group that you haven't heard somebody else bring forward. And then the next one is going to be a reactionary discussion round where we'll open it up for folks to have a little bit more of a dialogue around it. Any questions before we go into breakout rooms? April,